Hello students, this is Dr. Faizan Mirza. I'm discussing questions related to coordination and control. This question is from May, June 2019, variant 42. The diagram here, the figure for 1.1 is an electron micrograph showing a myelinated neuron. And if it's a myelinated neuron, you can see this cross section. This region must be the axon. And all these folds, these layers, these must be the Schwann cell which are wrapped around the axon to make the myelin sheath. This axon is either of the neuron which is not myelinated, or probably it is cut at the region of node of Renvier. Either could be the reason. So all these layers, these lines, these are the folds of the Schwann cell as it wrap itself around the membrane of the axon. So they are asking you to name the name A and B. So we know A is Schwann cell and the myelin sheath, and B is the axon. Explain what is meant by saltatory conduction and describe its effect on transmission of nerve impulses. We know saltatory conduction occurs on myelinated axons. We know the impulse is transmitted at a high pace. Local circuits they develop, and local circuits they develop at always between the across the adjacent node of Renviers. And as a result of this, the impulses start to jump from one node of Renvier to the next, and only these are the regions across which the ionic exchange will occur. And the areas which are covered with the Schwann cell, they lack membrane proteins as on the membrane of axon. So no repolarization occur across that. So the impulse jumps from one node to the next and then the next. As a result, the impulse keeps getting transmitted at a very fast pace. That's saltatory conduction. Then the next part is the type of sea snail. They mentioned the name. We know the first letter of the first word here represents the genus. The second word represents the species. It produces a toxin that block calcium ion channels in the presynaptic knob. We know, if you can recall, this is the synapse, and the synapse is having a presynaptic membrane. This A is the presynaptic membrane. This region C is the postsynaptic membrane. This region B is the synaptic cleft. We know that the vesicles are released from here and they come here and they cause the membrane to get depolarized. And as a result, this is how the neuromuscular junction, and this is how the synapse work. So they are saying that ion channels are blocked in the presynaptic knob of the cholinergic synapse. Cholinergic synapse means this synapse is releasing the acetylcholine neurotransmitter. The presence of this toxin results in no action potentials in the postsynaptic neuron, which means that the neuron is not getting depolarized. The postsynaptic neuron stays at resting membrane potential. It doesn't get depolarized. Explain why the presence of this toxin results in no action potentials in the postsynaptic neuron. So what's happening here? Basically, when the action potential arrive at the presynaptic membrane, the voltage gated calcium channels open, allowing facile diffusion of calcium ion into the presynaptic neuron. We know that calcium ion enter into the presynaptic neuron, and these calcium ion then trigger the release of neurotransmitter vesicles into the synaptic cleft. Now, since this toxin is there, this toxin will prevent the ion channels of calcium from opening. As a result, the neurotransmitter will, we know that neurotransmitter once released, it binds to the receptor in the postsynaptic membrane, it depolarizes it. But the presence of this toxin, the calcium ion will just not enter the presynaptic end, as that the neurotransmitter will not be released. And when doesn't get released, the receptor on the postsynaptic membrane would stay vacant and no action potential will be initiated. So that's how this will prevent the working of the postsynaptic cell. So this is how this particular toxin works. So let's take another question. This is from October, November 2018. This is variant 42. In this question, this is the muscle. This is the muscle shown to you. Electromicrograph of a striated muscle. Why do we call it a striated muscle? Because we can see the stripes here. We can see this is the Z line. This is the Z line, and this region in the center. This is the M line. This is the M line. This is the Z line. So, and this entire distance between two consecutive Z lines is the sarcomere, we can recall. And we know that these are thick filaments and these are the thin filaments. And these are the region of the overlap of thick and thin filaments. These organelles, these are mitochondria. How do we know these are mitochondria? Because if you look closely, we can see cristae here. And we can see the typical shape of the mitochondria as well. So these are all mitochondria present inside this particular striated muscle. Three structures have been labeled A, B, and C. A is the region with the mitochondria. B is the region where you are having the thick and thin filaments combined, as well as the myosins and the M line. Whereas C, this region is the actin alone, and this is the Z line. So the question that they are asking you is to label A, B, and C. You should use the letter. So where are the regions where myosin and actin are present? Of all these, so myosin and actin both are present in BC, because BC is B is the region, not C. B is the region where actin and myosin are present together, because this is the region of overlap. Then there's an M line, and then there's a region of overlap as well. Actin alone is present only in the region C because this is having the Z line. We know that the Z lines, all these lines are Z lines where the actins are held at a certain point in the center. They're asking ATP synthase and ATPase. These are the two enzymes that you have to keep in mind. ATP synthase is the enzyme that synthesizes ATP. It produces ATP, and ATPase breaks ATP. So keep that in mind. These are two different enzymes. They have the opposite functions. So A and B respectively will be we can place them for ATP synthase and ATPase because we know mitochondria produces ATP, and myosin head here is having the enzyme ATPase that breaks the ATP molecules so the cross bridges can break. Then they're asking to explain the role of ATP in contraction of striated muscle. We know that calcium actually triggers the release of acetylcholine as well, and calcium in the neurotransmitter in the presynaptic neuron. But in the striated muscle, calcium performs another function. It binds to troponin, and once troponin is attached with calcium, tropomyosin moves away. So when troponin and tropomyosin move away, binding site of myosin head on actin gets exposed. When the binding site of the myosin head on the actin gets exposed, myosin head jumps to bind the actin and tilt inwards, pulling actin inwards towards the M line of sarcomere. The cross bridges are broken, the cross bridges are made, and they shorten the sarcomere by 10 nanometer. That's one part stroke. The cross bridges then start to break, and they get broken because of hydrolysis of ATP. Because we have to explain the role of ATP here. So ATP hydrolysis allow the cross bridge to break. The myosin head is having ATPase enzyme, which we already have mentioned in the previous diagram accordingly as well in the table. That this enzyme will break ATP. Myosin head will then detach from actin. ATP is also used, apart from this, it is also used for pushing the calcium ions back from the sarcoplasm into the sarcoplasmic reticulum once the contraction is ended. Here, taking this another question from May June 2018, variant 42. This question number 8. Describe how the annexation potential arriving at presynaptic neuron can result in the release of acetylcholine into the synaptic cleft. We just need to describe the mechanisms here, and this question back is based on facts. Recall what's happening in the presynaptic membrane. It becomes depolarizes. Calcium ions they enter from the voltage-gated calcium channels because of the calcium and flux. The presynaptic neuron allow the acetylcholine to get released. The vesicles they move out by exocytosis. They use energy from ATP while exocytosis occur, and acetylcholine molecules then are released into the synaptic cleft. And once in the synaptic cleft, they can carry on the procedure further by binding to the postsynaptic membrane. Lidocaine is a local anesthetic drug which blocks voltage-gated sodium channels and postsynaptic membrane of synapse. Explain the effect of lidocaine on the action of postsynaptic neuron. So we know that if this question is an extension question. So they have already mentioned a drug lidocaine, and it's an anesthetic drug. So what is it doing? So sodium ions will not be able to enter the postsynaptic neuron. The postsynaptic membrane does not get depolarized to threshold. Action potentials are not generated, and the impulse is not transmitted because lidocaine will keep on blocking the voltage-gated calcium channel in the postsynaptic membrane of the synapse. This diagram C here shows neuromuscular junction. You can see there's the presynaptic neuron here, motor neuron. This is the Schwann cell. This is the mitochondria. These are the synaptic vesicles here. The gap here is called as neuromuscular junction or the synaptic cleft. So this B membrane is a sarcolemma. So you have to label myelin sheath, sarcolemma, mitochondria accordingly. Then they are asking you outline the importance of structure A in the transmission of impulses. Structure A is the myelin sheath. So you need to just outline the details of the saltatory conduction, that how neurotransmitter, how the not the neurotransmitter, how the Schwann cells they form myelin sheath, and the node of Henry are the only region across which the impulse keeps on jumping. Local circuits are developed across adjacent node of Renvier. This speeds up the neurotransmission, the nervous transmission, and this is called saltatory conduction. Explain why the structural labels C are needed for neuromuscular junction. C is the mitochondria. So you can see they link mitochondria with this topic often. So mitochondria are the site of aerobic respiration. They provide ATP. We know that ATP is used for maintaining RMP through sodium potassium pump, and they are present in the presynaptic membrane as a postsynaptic membrane because the sodium potassium pump is required at both. ATP is also used for synthesis of acetylcholine, yes, and packaging it into vesicles because acetylcholine molecule will be needing ATP for synthesis and for storage. ATP is also used during the exocytosis of vesicles. You can just describe this particular detail. That's it, I guess, from my side for these three questions. Thank you so much.